Good. So you should be able to hear us online. And thank you for coming today to the CGGG. So the speaker of today is Meenu Bhati. She's from India. She did a bachelor in veterinary science and from CVAS, Bikaner, India. And she did a master with research track in animal breeding and genomics at Wageningen in the Netherlands, and her PhD in animal genomics from ETH in Zurich. And she will have her defense soon, so it will be soon Dr. Meenu. And on the characterization of genomics variation and its association with complex traits and gene expression in cattle. She is currently a research fellow at the Roslin Institute in the Tennessee lab. So Meenu, the floor is yours. Everybody should be able to see your slides now, and you have about 45 minutes.
Thank you very much, Clemens, and thank you everyone for joining me for this presentation. This work is my doctoral study, the final paper of my doctoral studies, and we work on structural variation and short tandem repeats and how they are affecting gene expression and splicing. And this was supervised by my PhD supervisor, Hubert Pausch. So starting with this, what is the short tandem repeats first? So the short tandem repeats are the small repeat units which are repeated several times in the reference genome. And in this example, you can see this is a 3 base pair motif. As a reference sequence, you call it motif. So it's AGC, which is repeated 3 times in the reference genome. And when you take another animal genome or any other genome, and sometimes you find that there is insertion. So here you could see that one repeat unit AGC is added again. So it's an expansion of tandem repeat. You can also call it an insertion of repeat unit, but it's not always insertion. It could be a deletion of one repeat unit. It could be addition of some kind of a SNP inside these tandem repeats. So all together, these variations are called short tandem repeats when they are repeated several times, and usually the motif size vary from 1 to 6 base pair. And next part, so what are the structural variants are? So these are mainly the genetic variants which are larger than 50 base pair, which could be which could be divided into multiple of these different varieties, such as inversion, when the sequence is inverted within the genome, translocation, movement of sequence from one chromosome to another, deletion, when a part of that is deleted, duplication, when exactly same sequence is repeated, either in consecutive manner and non-consecutive manner, and insertions, when new additional sequences are inserted. And apart from these, other structural variation could be a combination of these multiple events. And when you cannot identify them precisely, you call them band, because you do not know where the other mates actually precisely mate. So here you could see that on chromosome 13, there are 2 kind of a mate or and are identified, but those 2 are aligned somewhere in a contig 1. But since we never look at those ones, so they identify, and this was inserted from context. So usually we do not identify them, and they come as a band in the VCF format. But those 2 breakpoints are still interesting to see what is happening. So in this paper, other than STR, we mainly focus on inversions, deletion, duplication, and band. Insertion and translocations are not part of the study due to the methodological stuff. So why we are interested in these genetic variants? One of the main reasons we are interested is that they are found to be associated with the complex trait, disease, and molecular phenotypes such as gene expression, splicing. And there are several studies in human who identify that how they could be associated and how they are actually impacting a lot of expression and phenotype. And not only in human, there are also some studies in cattle who identify that these genetic variants are associated with the phenotype, such as 660 kb deletion in the livestock related to fertility. Another one recently identified 12 kb multiple copy number variation, and more recent study who are talking about how the tandem repeats are associated with splicing variation. Although there aren't much study in cattle by far who have investigated their effect altogether on the expression level. So the idea of this paper to understand this part, and to do that, in our paper, we followed this basic concept. So we had the whole genome sequence data of 183 animal from 5 breeds and their crosses mostly. And we identify the reference base STR and then genotype them with the hipster tool and further filter them to identify very high quality variants. And for structural variation, we use the SMOO pipeline, which is using lumpy SV typer and dufold to identify very reliable structural variation, which are also further filtered based on depth, quality, and precision points. And here, out of those 183 animals, the sequencing coverage varies a lot from 5x to 30x. But the 5x animal are the publicly downloaded database, publicly downloaded whole genome sequence from Holstein and Flackley. But we keep that just to see how it looks like. And out of those 183 animals, we have for 75 animals, we had the RNA-seq data of which are paired and total stranded total RNA-seq. So we conducted from a part of the subset on this animal, perform eQTL and sQTL study on 75 animals. And all these study we perform for RNA-seq, we align the RNA-seq data with the star and further filter them with WASP. And we quantify with the QTL tools and feature counts. And finally, the splicing quantification was done with RAG tools and leaf cutter. So we had the phenotype data from the molecular phenotype, and we performed using the QTL tool cis. We did not focus on trans here. So here we did the 5 sets. So one, we only run this analysis with SNP and indels together, which usually everybody do. Next, we did it only with STR, other with SVs, and next with SV and STR. And finally, we combined all these genetic variants as a joint all to see how they are. And finally, we discussed the mainly properties when you contribute SV and STR. And why we only choose this one, I will explain. So what do we get first? So when we run the tandem repeat finder on the reference genome, we identified almost 1.2 million short tandem repeats, varies from 1 to 6 base pair, and that encompassed total 24.9 MB of autosomal sequence. We excluded as X and Y. And among these 1.2 millions, more than 99% of STR's total length was below 100 base pair, which makes us use the short read data because our short read data alignment was 150 base pair. So when the STR is below the size of your read, it is easy to identify them. And in this reference base STR, mostly we identify the sequences with the one, most frequent was 1 motif base pair size and the least with the 6. And in this one, when we genotyped them in our 183 samples, out of 1.2 million, we finally filtered 374,000. You would ask why this number is so less. So out of 1.2 million, we identified 800K STR sites, but not all of them were segregating in the population because hipster also identified those variations which are not segregating. We filter out them. Further, we did extra filtration based on the quality metrics which was suggested. And then we ended up finally with 374,000. And out of those ones, 71.1% were actually multi-allelic. And here you can see that, again, they are following similar to the reference STR distribution, the least observed on the 6, and the most observed on the 1 STR motif size. And these proportions, if we check which part of the genome based on annotation they are aligned, so you could see that mostly are intergenic. Pink is the intergenic, and orange is the intronic. Pardon me, I forgot to put the annotation scale. So all these polymorphic STR mostly were present in intergenic and intronic sites. And when we count the number of STRs along the animals, they vary from 73,000 to 189,000 per genome. So the least was observed with the least coverage, and the highest was observed with the highest coverage, and the higher insert size. And here you could see in these polymorphic side, as I said, they are multi-allelic. So you could see that here 90K sites which have only one alternative allele, rest has a very high number. And among these, when we check what are the actually motif-based sequence in the in per STR motif. So here you could see mostly A and T in the when the motif size. So you can only have 4 A, C, T, G, but it's only mainly T and A, not C and G was observed. And this was intriguing because then we tried to check in the human. They observed the same, that G and C are least observed. And this could be ideally because of either sequencing, or it could be that they are more, how would I say, they are less tolerant for the having more variation in them compared to A and T. And in two, so mostly it's GTAC and 3 in AGC. So these are the most common frequent motifs which are observed in the cattle, and we observed almost similar in the human as well. Further, for structural variants, we identified 19,000. So out of 50,000, we finally filtered and left with the 19,000 after high quality check. And these are also vary from 4,000 to 6,000 along the different cattle genome. And similar to STRs, they were also observed mostly in intergenic and intronic. And you could see we observed most likely the deletion. And there is also previous research study also say that when you use the short read sequences, their chances are higher you will observe deletion more than the other variation. And among these ones, we identify more number of bands, but since they have two mates, they are double in number. As I was mentioning with the coverage and insert size effect, so here you can see that the coverage in the genome, this is the breed-wise distribution. So here you could see that along with the coverage, the number of SVs are increasing, but interestingly, you will find this cluster and you would say why the number is too less, even the coverage is high. The reason was that the insert size, when you split a DNA into a multiple chunks and you try to sequence them, there is an insert size, means insert size is equal to the read, both the paired read and the distance between the both read pairs. When you have a very less distance between the both read pairs, so the insert size is small, then usually you identify less number of structural variation, and that was the reason, because they were almost similar size to what was the read depth, we observed less number. So it makes also sense if you have a large insert size, chances of identifying more structural variation are also high. And with these, we also check how are their size distribution. So you could see that mostly duplications are below 1 kb, whereas deletions are a bit more higher than 1 kb in the size. Whereas we find some of the inversions were extremely long in our data, and we tried to check it by ourselves as well with the IGV, and most of them were reliable. So we keep only the reliable inversions in our data. And band, I did not see because they all were one base pair in unit. Next, once we identify STR and SV, how they are present in the genome, we wanted to check the LD between SNP, STR and SV with the surrounding SNP and indels. But the one problem with the STR is that they are multi-allelic. So what do I mean by that? So here again the example. In my VCF file, you could see that this was the 80 motif, which was repeated 3 times, but I identified 2 alternative allele. One has a 5 repeat unit and one has 4 repeat unit. So in the VCF file, you will find a extra tag which is called BP difference, which is base pair difference. So if I compare this sequence with this, so the base pair difference is 4. For alternative allele 2, I have base pair difference 2. So imagine a scenario, I have animal 1 in which the genotype is reference and alternative 1. So GB is one of the tag which just put the base pair difference with the GT. So because for alternative allele, the base pair difference was 4, what we did, we just sum these value of GB and we call them a dosage. And if you call them a mean repeat unit, so we just take the mean of both the number of repeat units. So here it's 4, which is 3 and plus 5 divided by 2, 4. And so basically we use these dosage score to say that to identify the LD with the SNP. So this kind of LD identification is called the length base LD calculation for STR. There is another way when you split the STR in a multiple allele unit, and you can also calculate that, but then your LD will also decrease. There is a paper from Gimrick et al. In Nature Communication where they explain these on the imputation reference panel. And apart from, so we stick with the length base dosage. And for STR, SNP, and indels, as we all know that we always have the genotype in 0, 1, 2. And we calculated the squared Pearson's correlation coefficient between the 50 kb up and down of these genetic variants in each unit. And here you could see that mostly it's the deletion and STR. So more than 40% of the deletion and STR could be tagged with the SNP and indels, which are in a very high LD, like my approximate is 0.8. But you could see that band duplications and inversions, they are very difficult to genotype, to tag with the SNP and LD, which could be a multiple reason why it's there. First of all, band, they are the break end units, which could not be very reliable and not correctly identifying which kind of structural variance is there. It could be a large insertions which are not there, so properly not tagged. Similar with the duplications as well. Sometimes duplications are also in terms of copy number variation, but to identify that, you would need a very high coverage data to say that how many number of copies are actually replicated in duplications. So these could be one of the reasons that they are not properly tagged. Further, once we know about how's the structure in our data, we performed eQTL and sQTL study. We kept the 19,000 genes and 76,000 intron cluster after filtering them. And out of those 75 animals, we have only 12,000 structural variation, 271,000 STR, and 13 million SNPs and indels left, which were filtered after 0.05 MAF. So we want to check how the common variants actually behave, not the rare, because our sample size was not high. For the STR, we did extra filter because there could be an outlier problem because of the multi-allelic sites. So what we did, we only keep those genotypes which were at least observed. So if a genotype is observed at least in 2 animals, we keep that one, and other we replace them with the missing. And then we only keep those sites which were at least 80% genotyped. And we perform eQTL and sQTL. Another problem which comes that when you use the QTL tools, it does not take the multi-allelic sites into consideration. For that one, we identify if we check the C++ code of the QTL tools, if you replace the alt site with just one thing, it will consider because they count how many number of alternate alleles are there. So we replace them with STR, and QTL tools can use dosage. So rather than using the genotype, we use the dosage, which providing with the DS. And you need to make sure when you put them that they are in an integer form, not in the hashtag on the VCF file, because QTL tools also take that into consideration, what is in the header. In the first eQTL output, when we performed when we included all of them, we identify at least 34% of the E gene which are associated with the multiple E variants. So 25 structural variation, 514 STRs, and 5,000 SNPs and indels. When we did the Fisher test and odds ratio to check, and we find out they are 3-fold enriched if you compare with the SNP and indels which are present on the whole genome level in the all eQTL database, all eQTL analysis. And if you check this one, so because we performed this analysis on every set, so here you could see that you would find almost all genes, most of the genes, which are 3,000 E genes, are observed among all studies except the structural variation. And next, they are observed in again, but there are quite high number, 1,400 genes only observed between SNP and joint all. So this could also state that a lot of gene variation are all only associated with SNPs, or it could be that we are also missing a lot of ASVs and STR, which are not genotype, which could be significant as well. But it's difficult to say on this point. But that's the interpretation from our first results. And further, we only, as I said, we only took the property of SV and STR, because if I only do a enrichment and only analysis on 25 SVs and STRs, it might be that I'm missing a lot of short tandem repeats and structural variation, which could be likely causal, but missing because of not proper genotyping or alignment error or by any kind of issue despite of high filtering. So in the only ST-SVR analysis, the first one, you could see that if you check how much percent proportion of were actually, you could see that deletion, inversions, and the STR has way more larger proportion, which are associated with the E genes or as EQTL. And both of them are also associated with the very high number of, multiple number of E genes comparative to the SNP and indels. And if we check comparative to the E gene, how much they are distance, mostly are present within the 250 kb of the distance of the E gene. And among, okay, sorry. There it is. Okay, it works, thanks. And among these one, we also check how many are actually were overlapping with exonic E genes, how many are overlapping with, which are overlapping with genes but not E genes. So they are called exonic others. So here you could see that among the duplications, which were not so high but still significant. Okay. Sorry.
Yeah.
Okay. Okay. So, so when we calculate the Fisher exact test based on all variation, how much they are, we identify that deletion and duplications mostly are enriched in the exonic regions of E gene. And not only that, they are also increasing the expression, duplication is increasing the, which are present in exonic, increasing the expression of gene, which also correct based on how we assume if a gene is duplicated, chances of them higher that they will be expressed more. And this could be because of multiple mechanisms. So either the whole gene is because the whole gene duplicated, or other splicing, which also increase the expression of these variants. And whereas when we check the STRs, they were actually depleted in exonic regions and very enriched in the intronic regions. And this proportion is actually less than what we observed with the intergenic sequences. If we check the whole genome level, it was the intergenic STR 10% more than the intronic, but we check when the EQTL, intronic has more larger proportion. And when we check the LD again among this subset of 75 animals, and we were very interesting to see that most of the identified EQTL, especially deletions and STR, were in a very high LD with the surrounding SNPs, whereas this was not repeated for the band inversions, which is same as expected what we see on in the reference panel. So the example of, I'm talking about so many STR and SV, what they did. So this is one of the candidate example we tried to find. So here for this TT7B gene, you can see that this is a STR, which was a replicate of a GT motif repeated 11 times in the genome, and it's present on the 7th intron of this gene. And if you can see the normalized expression of this gene, which is divided based on the mean repeat number, as I explained before, and you could see that on the top, this is the number of animals which we observed in our data set. You could see that they are sequentially increasing. And this is also explained by our EQTL top analysis results, that this is among the top E candidate. Similar, also in the structural variation, you could see that this deletion, this is encompassing the whole novel gene, and it's downstream of this SLC gene. And this deletion was causing decrease in the expression of both the genes and among the topmost candidate in the eQTL analysis. So at least it suggests that yes, they are significant and they should be taken into account when we perform any kind of association studies. Further, after doing the eQTL, we also check for the splice if they have any effect on the splicing as well. And we find out similar results what we observed with the eQTL, that they are also not threefold, but more than twofold enriched. And but interesting thing in the SQTL that we identified the intron cluster, as I presented before, 76,000 intron cluster, but not all of them actually overlapped with the genes. Only 58% precise boundaries of introns overlap with the genes. So it also saying that our annotation is incomplete and there are a lot of transcripts which are present in the data set, which are not present in the annotation and should be taken into account, because leaf cutter does not take the annotation into account but also identify other splicing junctions. So rather than performing it on a gene level, I perform the whole analysis only on the intron cluster and just check how many overlap. And here you could see that the number of intron cluster, it's very similar to what we observe with the E genes. It's also shared a lot among SNPs, SVSTR, and joint all, and a lot observed only with the SNP, and very less with the SVs, which are sometimes only identified by them. And similar to that, similar to eQTL, here we identify that these STRs and SV are even more closer to the splice junctions, like only present within the 100 kb of the in the data. And among these, mostly is the deletion and STR, which were associated with the multiple intron clusters, which could be part of the same gene or multiple gene. And when we also performed the enrichment analysis on different annotation category, we observed this time that deletions and duplications both are enriched in exonic regions. And again, STR were depleted in exonic, but enriched in intronic region. So it's explaining that STR are really significant when it comes to intron and could be likely associated with the molecular phenotypes. And here you could see that if I check the effect size, so the duplication is still both positive, whereas all of them observed a bimodal effect. It means you could also identify positive and negative effect on the splicing. And again, interestingly, majority of our deletions and STR were in very high LD. As I mentioned in the first, overall on genome level, it was only approx 40% were in high LD. So it's likely that only those were in a high LD were part of this panel, which were identified with the eQTL and SQTL. And similar to eQTL, we also try to check the candidate SQTL. Here you could see one amazing thing, do not ask me why, both are present in the upstream of the gene. So this STR, we identify the upstream of this long non-coding RNA. But if you see these green boundaries are actually the splice junctions. And this splice junctions were highly associated with the splicing of this long non-coding RNA, which you can see from the intron excision ratio. And it's saying when the number of repeat unit increases, the splicing increases. But if we see the expression level, it's totally opposite. When we checked, we also realized because our this intron boundaries were not part of the actual annotation. So it's likely that our quantification did not even measure this new transcript, which was probably available, but not in the annotation. So we did not find any difference. And this was among the top candidate. And another example of the structural variation, this was also present in upstream of, this deletion was also present upstream of this CAP1 gene. But here you can see that the upstream was, this deletion was actually causing the decrease in the expression of splicing ratio of this particular intron cluster, which again, this was not fully overlapped with this gene. As you can see that one of the splicing junction is present outside. So overall, SQTL is telling that a lot of transcripts which are not available should be identified and could be add-on in our study. And these variation does affect significantly on this. So I hope I did not take much time, 30 minutes. So overall conclusions that in our study we identified that STR are mostly multi-allelic in cattle and they are predominantly non-coding, and spatially intronics are spatially enriched in the intergenic and intronic. And the number of STR and SVR are higher if your coverage is high and also if you have a large insert size in your data set. And next, both ST and SVRs could be, are actually more than twofold enriched as a splicing and EQTL, and they both are associated with the expression of multiple genes. And this could be further even checked with the multiomics data, which we don't have on these samples. And finally, these exonic and splicing duplication and deletions are enriched in exonic regions, whereas STR are enriched in intronic regions, which could be taken into account for future studies to include these panel in the study to identify if they are associated with it. Thank you very much. And with this one, I would like to acknowledge my professor, Hubert Pausch, Zina, my co-authors, and Odal, and the whole animal genomics team. The paper is out, at least submitted, finally. And I would also like to acknowledge all the funding acquired by Professor Hubert Pausch with the Swiss Genetics previous one from the ETH funding, which was very useful to conduct this study. Thank you very much.
Do we have any questions in the room? Then you need to shout so people online can hear you. If you don't hear the question online, just pop a question on the chat. Well, say it on the chat and then we'll repeat the questions in the room. And if you have questions online, you can pop them in the chat or just unmute yourself and ask questions. So, any questions in the room?
Hi, Meenu. Thanks very much. It was interesting when you saw that the LD pattern was different for the different types of structural variants.
Yeah.
So is there a biological reason why that would be the case? And did you check the Hardy-Weinberg, whether they were in different, so did you check whether they were in Hardy-Weinberg equilibrium, these different types of structural variants, and did that differ between the classes?
No, I did not check that because I keep them filter for more than 0.05 MAF, so I was expecting that they would be still segregating nicely. I did not check that Hardy-Weinberg, but would be good to check why they were not in a high LD with the other stuff. But one another thing from another paper, one of this Science or Nature paper on the structural variation, they mentioned that one of the reason, because they had this data set, they said these variations were largely present in the largely present in those segmental duplications and complex variants quite close by. That could be one of the reasons that they are low in LD with these variants, and we did not have that data to check in our data set if that could be a true case or if they are overlapping, because we actually excluded any regions which were present in a which were any part of a reference gap. If there is a reference gap, there shouldn't be any SVs around that because they would be likely false because of the alignment issues. So we also did not take those ones. So that could be one of the things, but I will check the Hardy-Weinberg if they are. Yes, sir?
It's more to James. I don't know much about this. So I will apologize before I open my mouth in case it's rubbish. You said you had crosses. Normally you don't expect crosses to have the Hardy-Weinberg equilibrium in terms of allelic frequencies. But would you biologically expect that to happen when you are looking for these SVs, or does it follow the same norm?
Yeah, I guess I was meaning more, have you compared the distribution of Hardy-Weinberg between the different structural variant types? So I take your point that if they're not randomly mating influence either populations, then they're not necessarily going to be in Hardy-Weinberg. But I would have thought you would, if you match by other frequencies, they should show a similar distribution of Hardy-Weinberg across these categories. If they're falling out of Hardy-Weinberg disproportionately, I guess it may suggest that maybe there's more false positives in there, because some of these are harder to call than others. And does that help explain your healthy pattern? Because maybe you've got a different number of false positives in one category than another. It's just a possibility, just to maybe rule it out.
Yeah, that's a good thing to check for the next review.
Yeah, I was also a bit wondering about the population structure. I mean, those examples of differences in expression or the different numbers of repeats or whatever.
Yeah.
Can you know that those aren't just associated with a particular founder breed, because you've got quite a few different breeds in there?
Yeah, in the total
rather
panel.
than, in other words, is it a real effect, or is it something to do with the genetic background of the different breeds that you might have?
For the LD pattern in the whole,
No,
or on
for example, you have differences in expression related to a marker, which is the tandem repeat number, but could that be associated with the breed that it came from, rather than the than being something acting very locally?
Yeah, for this one I can say that all this sample of the RNA-seq, they all were part of the brown Swiss and OB crosses, and both breeds are very closely associated. So they separated in 150 years before.
Okay, so it's not that whole population where you had
No,
all sorts of.
it's a very close cohort, which was very close. So idea was to keep. My colleague did the whole selecting those animals which look like brown, and identifying which are actually their breeds are. So they try to keep the cohort on a very close level, so the breed differences does not exist. But the breed differences does exist in the whole population level, but we did not differentiate on the breed level, because it's not a justice when you have one coverage on a less than 10 and other you have 30, and you are saying that, oh, you identify more there, it wouldn't be reliable. But it's still good to see that, because you would capture some variation which are not captured in other breeds.
Any questions online?